Okay, I think just, I guess, in the interest of finishing on time, I should probably get started. So welcome. Hopefully this is the breakout room that you are hoping to be in. By no means feel obligated to stay for the entire duration of this workshop. You can come and go as you please. And also, since we're a small group, at least right now, please feel free to interrupt me if you have any questions along the way. Or if you think there are bigger discussion points, then you can save them until the end. So I've included my email address here as well, if you'd like to follow up personally after the workshop is finished. So what I'm hoping to cover today is quite ambitious, but it's outlined on this slide. And so I'd like to talk about the optimized measurement of perturbagen response, essentially. And so I'm going to walk you through the motivation, and this is essentially why do we need GR metrics, as well as what they are. The methods that we use to collect dose response data typically in our linked center, and these are DyeDrop assays. So I'll cover both the experimental techniques and some computational tools that we've made available to the community. And then if time permits at the end, I'll just touch upon some, I think, important takeaway messages, as well as a few lessons that we learned from the multi-center links collaboration that Peter referenced in his presentation this morning. So jumping right into the motivation, typically when people measure dose responses in cancer research, they look at the relative cell number. This is usually looking at the number of cells you have at the end of an experiment, and you look at that relative to an untreated control to generate dose response curves that look something like this. So you can take one cell line, treat it with a number of different drugs. A drug that is not as effective or not as potent will be further to the right on these dose responses. A better drug will be towards the left. The same curves could just as easily be for different cell lines with one drug. And so here, instead of looking at the effects of two different drugs, you'd be looking for resistant and sensitive cell populations. And so this slide will be familiar to those of you who sat through Peter's talk this morning. Just looking at conventional drug response metrics in cancer research, or really in any field, to make the point that although IC50 has typically been what most people use and most people reference and most people seek out to make comparisons between different drugs and different cell lines, there's actually a lot of variability in all of the different summary statistics for these dose response curves. So that includes the Emax, looking at efficacy, as well as the shape of the hill slope for these sigmoid curves. And so about 5 years ago, there were a number of papers that came out that called into question some inconsistencies that were observed between some very large pharmacogenomics studies, looking at many cell lines and many drugs. And we've highlighted one here, but there's a whole list of references that address some of these concerns, or at least bring them to the I guess announce them essentially to the community. And so to start to try to understand what may be underlying some of these differences, it's important to kind of deconstruct a drug response measurement. And so what exactly are we doing? In our lab, we do everything microscopy based. This obviously isn't the only way to do dose response studies, but I think I'll argue as we proceed through this workshop that there are a lot of benefits to doing microscopy-based dose response studies when possible. And so when you're collecting dose response data, you're starting with cell population. If it is untreated, will hopefully grow exponentially over the duration of your experiment. If you treat it with a drug, in this case a cytostatic drug, you'll see that there is no change in the number of cells between the time that you add the drug and the time that you measure the effect at the endpoint that you selected. If you instead look at a toxic drug, you're going to see a cytotoxic effect in that you have fewer cells at endpoint than you did at the beginning of your experiment. The images on the right are just representative images of what some of these populations might look like. And so you can collect these data at multiple different doses to then generate a dose response curve. You can fit it oops, yeah, there we go. You can fit it to generate some summary statistics, including that IC50 value that is so commonly reported in the literature these days. You can then collect similar responses, either for many drugs or many cell lines, and start to make or start to collect data on which are more potent or more sensitive, depending on what is interesting biology for your project or your context. And so if we take a closer look now and start to think about what's happening with cell lines that have different division times, which for any of you who are wet lab biologists like me, you can appreciate that having cells in culture, they have wildly different division times. Sometimes you need to passage cells every 2 days. Sometimes maybe you only need to passage your cells once a week or even less. Um, and so as you can imagine, this is probably a confounding factor when we start to look at relative drug effects. And so here we have a cell line that is growing relatively quickly. We treat it with a cytostatic drug, and after, in this case, let's say 3 days, we measure the relative, we measure the ratio between the number of cells in the untreated population and the treated population. Now we take a second cell line, it's growing considerably slower. We treat it with the same drug. We get the same phenotypic effect, which is one of cytostasis. So there's no net change. We do the same measurement and we look at the ratio of cells between the untreated population and the treated population, and we come out with a different ratio. And so if we take this example and we consider a second drug now, same thing, we're looking at one cell line that's growing quickly, one cell line that's growing, or two cell lines that are growing at around this, no, two, sorry, two cell lines that are growing at different rates. So again, the cell line that's growing quickly treated with that same cytostatic drug, and a second cell line that now is growing a little bit more slowly. And in order to come out with a ratio that matches the first cell line of 0.4, we actually have to dip into cytotoxic space. And so I hope that the problem is really clear from these two examples, that in this case, although you have the same relative cell readout, a ratio of 0.4, you have vastly different phenotypes and really important differences in the underlying biology between these two cases. And so what Mark Hafner, pictured in the bottom right-hand side of this slide, did was come up with normalized growth rate inhibition values or metrics. And so what he proposed is that we should actually be comparing the growth rates between an untreated population and the treated population, rather than just looking at a snapshot of how many cells do you have at endpoint. And so from a practical standpoint, you don't really have to do anything particularly different when you're running your experiment. So you still measure the number of cells you have in your untreated population, you still measure the number of cells you have in your treated population at endpoint. What you add on top of all of this is the number of cells that you have at the time of drug addition. So this is what I'll refer probably kind of subconsciously to as our time zero count. So if you know the number of cells you're starting with, you can then calculate the growth rates for both your untreated and treated population and compare those rather than just comparing your relative cell counts at endpoint. And so what this does is it gives you dose response values that are independent of division rate. We limit them from 1 to -1. Values between 1 and 0 are indicative of partial inhibition. A value of 1 means that there's no discernible difference between your treatment and your untreated control. A value of 0 is a purely cytostatic response, one in which the number of cells remained constant from the time that you added your perturbagen and your endpoint when you measure the number of cells that you have at the end of your experiment. Values below 0 are indicative of a net cytotoxic effect, and so this means that you're actually inducing some cell killing and you have fewer cells at endpoint than you had at the beginning of your experiment. So to give you an idea of what some real data look like, this is an example of docetaxel responses across a large panel of breast cancer cell lines. And what you can appreciate here, and I think is quite intuitive and part of what makes GR metrics so attractive, is just that there's a broad range of responses that vary from no effect up at the top to some cytostatic effects to frank toxicity in the more sensitive cell lines. And that in addition to seeing broad variation in the efficacy of docetaxel across breast cancer cell lines, we also see a really broad range of potencies. And so this is captured by looking at the GR50 value, which is essentially equivalent, at least conceptually, to the IC50 value of a conventional dose response curve. And so to just give another few examples, looking at a couple of drugs that have very different phenotypic outcomes, different mechanisms of action. If we look at the CDK4/6 inhibitor, palbociclib, you can see what you would expect to see for a drug that induces cell cycle arrest in G1, either no effect, and this is for genetic reasons, or cytostatic effects ranging from partial inhibition all the way to complete growth arrest. The CHEK1/2 inhibitor AZD7762 is similar to the docetaxel example I presented previously, where we see a really broad range in both potency and efficacy across a panel of various breast cancer cell lines. And then the third example that I'm highlighting here is that of dinaciclib, in which the potency is essentially the same across all cell lines. They're all about equally sensitive, or you see that kind of drop in the dose response curve at the same, approximately the same concentration across all cell lines, regardless of subtype. And the responses are almost exclusively cytotoxic, although the degree of cytotoxicity, of course, varies, which is shown by the spread of values between 0 and below. Okay, so to give just another few examples, I guess, of real world data, does this really matter? I'd just like to highlight the effects of division time on the perceived sensitivity of cell lines to etoposide. And so here we have a doxycycline-inducible BRAF V600E mutation that, as you ramp up the concentration of dox, you decrease the division rate of the cell. So you get a much longer division times as you get higher BRAF V600E expression. And if you look at the response of these cell lines with the different expression levels of BRAF, what you see is that the cell line that is dividing the slowest appears to be the most resistant. And this is a general trend. We find that kind of systematically, not across all drugs, but across many drugs, that cell lines that are growing more slowly appear to be more resistant in general to a number of different agents. And this, of course, is not necessarily true. And so when you, I'm just moving things around my screen a little bit. So when you correct these data for GR values, or for growth rate inhibition metrics, what you see is that actually these cell lines are equivalently sensitive to challenge us with increasing concentrations of etoposide. Looking at the same data, but just presented a little bit differently to really kind of drive home that important point. If we look at the measured IC50 with increasing concentrations of dox or higher levels of BRAF or slower division times, you can see that the IC50 value appears to increase by more than 2 orders of magnitude. Same thing if you look at Emax, it looks like you get a much more, the drug is much more effective, in conditions in which the cells are growing more quickly. Again, looking at the GR corrected data, you can see that these are in reality just flat lines, and that the cell lines are equivalently sensitive to etoposide. And so just, I'm not going to spend any time on these slides. I just wanted to include them really for your reference to let you know that in addition to these kind of conventional GR metrics that we use for all our dose response data, there's also a time-dependent GR equivalent that can be used to look at how GR metrics vary over longer durations of assays. And essentially, you're just looking at different time windows as you move through your data sets. And we've also looked at decoupling the cytostatic and the cytotoxic components of dose responses. I'll talk about this a little bit as we dig into the next sections, but this is really just to highlight that behind any GR curve, you actually have 2 components that are that are contributing to the shape of the dose response. One being a static response and the other being a toxic response. And so you can imagine that any GR value is probably going to be a little bit of a mix of cell death, cell proliferation, and cell cycle arrest. And so by decoupling these, you can gain important insight into differences in mechanism of action between drugs that, at least on the surface, in terms of the shape of their GR curves, appear to be similar. This is one example that highlights that point where you have two cell lines, MCF7 and BT20. These are both breast cancer cell lines, where the GR curves to luminescib, which is an HSP90 inhibitor, appear to be very, very similar. And so they both result, at least in GR space, in values around 0, which is we interpret to be a cytostatic response. When you look at MCF7 cells, the cytostatic curve here is shown in blue, and you can see that that really explains the full shape of this dose response. There's really no contribution of toxicity to this dose response. Whereas if you look at BT20 cells, you actually see that the cytostatic curve kind of tapers off at a certain point, and actually at doses right around kind of that inflection point, we see some toxicity. And so it's a combination of cell death and probably slowly proliferating cells that results in a net effect of no cell gain, no cell loss, with increasing concentrations of the inhibitor. Okay, so to conclude the first section, I think the most important takeaways are really that GR metrics eliminate might be a strong word, but they certainly ameliorate confounders that act by cell division time, and so they can help remove bias that's introduced by working with cell lines that grow at vastly different rates. GR metrics, they can be extended to quantify time-dependent drug responses. We didn't dive into that, only touched on it. And then that last point that I made just before this slide, that GR curves can be decoupled into their toxic constituents. And finally, although we haven't talked about this yet, we'll get to it, but that GR metrics, we think, improve reproducibility in studies that rely on measuring growth inhibition, such as most pharmacogenomic studies that we do in preclinical cancer research. So I'll pause there just for a quick second. If anybody has any questions, let me know or stick them in the chat. Otherwise, I
have a question.
Yep.
This is Heidi.
Hi.
How are you, by imaging method, identifying cells that are arrested versus dead, but are still sitting on the plate?
Yeah, so I think the next section will address that question. Let me know if it doesn't, and I can revisit it. But yeah, I think the DyeDrop assays that I'm going to walk you through next should hopefully answer that question. Okay, so I'll move into the experimental methods that we typically use in the lab of systems pharmacology at HMS to conduct our dose response studies. I will, I think, probably spend the bulk of the time discussing experimental techniques, but I will touch on the computational tools as well. Okay, and so this is an overview of the experimental pipeline. It's all kind of interconnected a little bit. So we go from experimental design, which is done computationally, which spits out the drug treatment files, to actually collecting the data, to analyzing the data, and then to interpreting it with GR calculations. And so there's lots of things to consider before you start any kind of dose response study. And so things that are important to consider are the number of cell lines that you want to test. For this assay, it's important to determine if they're amenable to imaging. So broadly, this means, are they adherent? A little bit more detailed, are they adherent? Do they spread out nicely? Cells, obviously, that grow on top of each other and clump are difficult to segment. There are things you can do to make those cells a little bit more amenable to these assays. But typically, what we care about, or what we work with most easily, are cells that grow well in monolayers on tissue culture plastic. And so The other thing that obviously needs to be considered before you embark on any kind of large scale experiment is what's the appropriate seeding density. And so, one underlying assumption with GR calculations is that your cells are in log phase exponential growth over the duration of your experiment. And so, it's important to seed your cells at an appropriate density so that that assumption is actually true. And so the other thing to consider is how many drugs you want to screen or you want to evaluate. Are they DMSO soluble? This can determine how you administer them to your multi-well plates. How many dose points do you need? Ideally, you want to be able to fit a nice sigmoid curve to your data, and so we typically do 9-point dose responses. You can do more points, you can do less points, but I wouldn't go a whole lot lower than 6 or 7 concentrations. Um, it's important to know what the appropriate dose range is. Again, in order to get a good fit to your data, you need to have some concentrations that are ineffective and some concentrations that are maximally effective. And so you want to make sure that you cover that whole range if possible. How many time points do you want to test, or what's the appropriate time point? Again, this can be at times dictated either by how quickly your cells divide or by the known mechanisms of drug action. Do you know that your drug is going to kill all the cells it's going to kill in the first 24 hours, or is this going to be kind of a slow build to maximum effect over the course of several days? Um, yeah, I think I covered those points. Okay, so all of that can then help kind of inform your choice of multi-well plate. 96 or 384 well plates are typically what we use. The assay could be the assays could both be modified to fit either multi-well plates with fewer wells and larger surface areas or whatever, but typically 96 or 384 well plates are appropriate for this type of work. And so the final question that is also important is do you actually need to use the GR approach? And that really depends on how you answer the previous question. So it's not always required or recommended, but there are certain cases when it really is critical. And so cases in which it's not necessary to include that time zero plate and compute GR values are cases when either your phenotype is not related to cell growth, your untreated cells don't grow, and so if you have a population that's just going to sit on your plate indefinitely, there's no growth rate to look at, so, so they don't apply. And then the third case is a case in which maybe your drug mechanism of action is really 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 quick, and so the assay duration is too short to compute any kind of growth rate. So those are really the three cases in which the GR approach isn't necessary. Or if you're doing everything in one cell line and you know that the conditions that you're looking at in a single cell line are the same, and that untreated, same plate, same media, same day, all of those things, then your GR results are going to match a relative cell count assay. Okay, so in terms of experimental design, we have computational tools available. I've put the GitHub address up at the top, at where all of these tools are available. So they're all written in Jupyter notebooks. And for the experimental design component, what you do is you essentially feed in several CSV files that include important experimental parameters for what you're interested in looking at. And so there are examples of these scripts and these CSV files in the GitHub repository. Feel free to browse those and reach out if you have questions or feedback about them. But essentially, you input a input file that contains information about the agents you're interested in, the maximum concentration, the number of doses you want to look at, the number of replicates you want to look at, as well as your dilution scheme, and a plate ID file that contains information about the number of plates you want to treat. Those ideally should be barcoded, and what's in those plates in terms of cell line identifiers, as well as the time points that you're interested in querying. And so what's output from those is a metadata table that can be used downstream to merge with the output from the microscopy analysis, a treatment layout visualization that is just nice to confirm that you've generated a treatment plan that is what you think it is, and a treatment file. So depending on the automation that is available to you, this outputs a D300 treatment file, which is an HP digital drug dispenser that we rely on quite heavily for some of our dose response studies that don't include a ton of different drugs. Okay, so if you are screening a large number of compounds, it's obviously not feasible to treat. I could argue it's not feasible to treat cells in 384 well plates manually under most circumstances, but especially if you've got a large number of drugs that you're interested in. And so a recommendation is to generate library plates for these that can then be used for pin transfers. If you have a moderate number of drugs, this is an example of what the HPD 300 software looks like. It enables you to add drugs and randomize the treatment layout across multi-well plates. And this is the treatment file that is output programmatically by that experimental design notebook that I showed you just a couple slides ago. Okay, so there are important design steps that we think can improve reproducibility. These include randomization of the treatments, which you saw both on the pin transfer library plate and on the D300 treatment plate. Standardizing nomenclature as much as possible. As anybody who's kind of gone back and tried to piece together data from experiments that they ran number of years prior, you can appreciate that standardized nomenclature really makes going back and finding things a lot easier and interpreting them a lot easier, and just gives you more confidence, I think, in your data. And so as part of this, barcoding the plates can be tremendously helpful. Controlling for plate biases really goes hand in hand with the randomization point that I made just now, that if you spread your positive and negative controls across your treatment plates and randomize your conditions, you can really mitigate some of the systematic errors that might be present from either using automation or from edge effects and things like that if your treatment, if your endpoints are several days. And so in addition to these kind of design features, using robotics as much as possible is also very helpful, and excluding edge wells if you don't need to use all the possible wells on a multi-well plate. And so this is just that randomization point that I made, but I think it's always nice to visualize these things. This is an example of edge effects where after a couple of days in the incubator, the wells around the perimeter of a 384 well plate have significantly fewer cells than the wells in the center of a plate. This column bias and row bias can be pulled out and plotted, but again, just by randomizing treatments, you can really take this systematic error and essentially turn it into randomized error, and that gives you it helps with reproducibility in terms of comparing dose response results. Okay, so now it's time to actually run the experiment. We know what we want to do, and now we have to execute it. And so, as with most dose response studies, as I alluded to earlier as well, it's important to grow happy cells and to have them plated at appropriate densities in multi-well plates to ensure that they're going to be in log phase growth over the duration of your assay. We need to deliver drugs to these multi-well plates, stain and fix the cells, image them, and then extract quantitative feature data from the images. And so just to walk through each step really quickly, at least in terms of how we do it in our lab, there are, of course, lots of different ways to do all of these things. We use a Thermo Fisher multi-drop combi to plate our cell suspension in 384 well plates. The key here is to make sure that your cell suspension is well mixed so that the plates you seed at the beginning have approximately the same number of cells as the plate that you seed as the plates that you seed at the end. Again, automation here makes this really quick. It helps with reproducibility, and we highly recommend barcoding the plates if possible. And so at this point, consider the number of test plates to have but also if your plans are to compute GR metrics at the end, seed an extra time-zero plate for each cell line that you're studying. And so the cell seeding density, again, just to kind of visualize some of those points that I made, and I think these are points that every kind of bench biologist is told, but you never really look at. But this really just drives home the point that if you plate your cells too densely, they're not going to be an exponential growth, or if you plate them too sparsely, there's actually going to be a significant lag phase before they enter exponential growth. And so you want to avoid both of those kind of shoulder cases with your experiments, and hit a division rate that is going to be able to be maintained over your experiment as much as possible. And you want to do that, of course, because seeding density affects the number of divisions, and the number of divisions can bias conventional metrics. And although GR metrics correct for much of these issues, it's still, again, best to it's still best practice to keep your cells growing steadily over your experiment. Okay, so your cells are happy, they're plated, they're at a good density. You've let them adhere probably for 24, maybe 36 hours, or in some cases, depending on the cell type, a little bit longer or a little bit less time. And now it's time to deliver drugs. And so if you're working with drug libraries, then you're probably going to be using pin transfer robotics to deliver your dose response titrations to your cells. This facilitates reproducibility. It makes sharing things between groups really easy. But if you're working with a smaller drug set, you're probably, or if you're lucky and have access to a digital D300 drug dispenser, you can use it. Drugs, it performs best with drugs that are prepared in DMSO, but it can dispense aqueous solutions as well. We typically use it for pilot studies, as well as for some validation and follow-up experiments based on larger screens. Oh, and that's actually the point I meant to make here, is that drugs that can't be pinned because they're in aqueous solutions can be dispensed with the D300. Or if you're limited in access to these technologies, a good old-fashioned manual serial dilution can give you good dose coverage over many ranges, and if done carefully and thoroughly, I guess, with a multi-channel pipette, can also yield very good dose response data. And so after you've waited an appropriate amount of time, it's then time for your endpoint analysis. And so I'll introduce you to two assays. They're both variations on what we refer to as DyeDrop assays. That'll become clear in the next slide, what the meaning of that is. In the simplest form, we're just looking at live cells and dead cells, staining them with hooks, and a live dead red dye, which is visualized in blue, or a more phenotypically rich assay that also incorporates EDU for cells in S phase, as well as phosphohistone H3 for cells that are in M phase. And so in the lab, what we essentially do, visualized here, is one well in a multi-well plate. You've got your cells on the bottom with your media. We use OptiPrep, which is a density gradient media. So we're able to layer in small volumes of dense stain solution, followed by an even denser fixed solution. And then we aspirate that and replenish the liquid with PBS. And so what this enables us to do is to really reduce the working volume for our dyes, which can be expensive, as well as our fix. And it means that we're staining and fixing our cells before we're aspirating anything from the wells. And so this helps preserve those cells that maybe are loosely adherent because they're stressed out or dying, or because they're starting to divide. The deep dye drop variation is performed very similarly. So in addition to adding in a live dead dye prior to fixation, you're also adding in EDU, which is then incorporated into synthesizing, or DNA that's actively being synthesized in the S phase of cell cycle. they're fixed. Similar aspiration, dispense of PBS, and then we block, perform a click chemistry reaction to label the EDU, and then incubate the cells overnight with a phosphohistone H3 antibody, as well as with the Hooked dye. The advantage of including the Hooked dye in the overnight incubation is that it can saturate and then give an accurate measure of DNA content. Shorter incubations with Hooked don't give you as clean separation between G1 and G2 cells. We perform all the wash steps with an automated plate washer. Again, this is just, one of those kind of advantages to having access to robotics. It ensures that every well is treated equivalently, and so you're not worried about, accidentally putting your aspirator in harshly on a subset of wells and kind of biasing results that way. And so a lot of the motivation behind these experimental techniques is in minimizing washing. And you can say, well, is washing really that bad? And I can give you a really bad example and say that, yes, it can be. And so this is an example of cell loss as a result of multiple washes. Of course, this is an extreme case, but it drives home the point that if possible, washes should be minimized when you're performing dose response studies. And that can, of course, bias your results. Cells that are stressed out, as I alluded to earlier, are more likely to be washed away. And so then that can ultimately affect the relative cell counts between your untreated population and your treated cells. And so moving into image acquisition at this stage, the cells are stained and fixed. They're stable in the fridge for weeks. So imaging can be performed, kind of at any time in short order. They're also stable at room temperature for several days if you've got robotics and plate hotels, and those plates are just going to sit out while they wait for their turn to be imaged. So at least in our lab, we've used a couple of different microscopes. Historically, we used a PerkinElmer Operetta, and then more recently, we've moved this assay over to a Molecular Devices IXM-C system. Any high content, high throughput microscope is suitable for this kind of thing. Okay, so in terms of image acquisition, what are we doing? The most important thing here is to image as much of the well as possible. Ideally, you're imaging the entire contents of the well. And this is, again, because cells may settle in a corner of a well. And if you don't image the entire well, you might not capture a, oh, that's a cool feature. Thanks, Greg. 15 minutes left. So if you don't image the whole well, you might not capture a representative density of cells at endpoint. And so for the DyeDrop staining procedure that I outlined, we're capturing 2 channels. We're looking at Hooked and the live dead marker. For the deep DyeDrop version, in addition to Hooked and the live dead marker, we're also imaging EDU signal and phosphohistone H3 signal. And so now that we've collected our images, this should actually be highlighted, we need to perform some segmentation before we move into classifying the cells as either live or dead and into their respective phases of the cell cycle. And so nuclei are segmented based on their Hooked signal. We measure all of the other channel intensities within those nuclear masks, and then we classify the cells. But what we really do at this stage before classification is we output the feature data. So in these text or CSV files, each row is a cell and each column is an intensity feature. And so again, we have similar notebooks to those that I presented for the experimental design, in which the raw data can be analyzed. And so you input your raw feature data, you input that metadata file that was generated during the design component of the experiment. And what you get out are, at least for DyeDrop experiments, a normalized count table, your GR values, your summary GR metrics, so these from the fits of the GR curves, and some summary plots for visualization. And so taking a closer look at what's going on under the hood in this particular assay, we're essentially just looking at the distribution of live dead red signal intensities and setting a threshold above which cells are dead. And so from the live counts, we generate GR curves. From the dead counts, in concert with the live counts, we generate the fraction of dead cells. And so these are complementary views, and I'll present, I think on the next slide or in a couple slides, just kind of how important it is to include this death metric in addition to the GR values. This is just a visualization again of the data that's output from a DyeDrop experiment. And so this is that slide. We often get asked the question of, is it appropriate to just count live cells? In some cases, sure. If you're sure that your mechanism of action of interest is purely cytostatic, then counting live cells isn't going to add anything. But shown here is a scatter plot of the increase in fraction of cell death relative to GR value for one of our screening data sets. And the point I think I want to make is clearest by looking at GR values of 0. And so what I hope you can appreciate here is that a GR value of 0 can correspond to a purely cytostatic response in which there's no increased cell death, or a mixed response in which there's a high fraction of cell death, which obviously then has to be balanced by some proliferating population. And so looking similarly now at the deep DyeDrop data, again, we're just inputting that raw quantified single cell level data as well as the metadata from the design component. What's going on under the hood here is a little bit more complex. We're looking at gating the DNA content based on Hooke's intensity, and we're looking at gating for S-phase cells based on the EDU intensity. And so based on those two dyes, we can classify cells as being in the G1 phase of the cell cycle, the G2 phase, S phase. We classify these cells that have intermediate DNA content but no EDU intensity as being S phase dropout cells, in addition to what are usually thought of as apoptotic cells that have less DNA content than your cells in G1, and polyploid cells that have more DNA content than your cells in G2. If for whatever reason the automatic gating doesn't do a very good job on your cells, we've built in the option to adjust these gates manually, and that's what's shown here. That can then be saved and used for all your downstream analyses. We summarize these cell cycle plots in stacked bar graphs, and so here we're showing two drugs that induce a G1 arrest. G1 is shown in purple, S phase in red, G2 in green, and then the other populations in the other colors at the top. And what you can see here is that in both cases with increase in concentration, you get loss of S phase cells and an accumulation of G1 phase cells. Okay, and so here, in addition to the three tables that I presented as being output from the DyeDrop assay, you also get all the cell cycle fraction data for each cell line agent concentration condition. And in plots similar to those that I showed to convince you, hopefully, that including a death marker is important, the cell cycle information is similarly adding a lot on top of what you just get from a GR value. And so if we're doing really not a whole lot of additional work, you get a lot of additional information. Again, drawing your eye to GR values of 0, those can come about from drastically different cell cycle effects, either loss of cells in G1, gain of cells in G1. Obviously, if they're lost from G1, they're accumulating somewhere else. And then the same plot presented for the effect on the fraction of cells in S phase. The assay is flexible, and so I presented the assay the way that we conceived it initially, which includes a phosphohistone H3 marker to give confidence in the M phase population of the cell cycle. But if the biology you're interested in isn't is different, that antibody can be swapped out for anything else. And so these are just a few examples of antibodies that we use for specific use cases, looking at CDK4/6 inhibition and its effect on phospho-RB or on cell size using actin, looking at DNA damage in cells treated with etoposide using a 53BP1 antibody to score foci, or looking at apoptosis. In this case, we're looking at actinomycin D treatments with a cytochrome C antibody that is released from the mitochondrial from the mitochondria in apoptotic cells. The assay is also amenable to further multiplexing, and so if you're interested in layering on additional antibodies in a cyclic way, that's possible. The click chemistry reaction either has to be saved until the very end or it needs to be quenched. The EDTA will do the trick. Otherwise, the copper component of the click chemistry will react with the bleach solution and obliterate your cells. So a couple little tricks, but it's totally feasible to multiplex these assays with cyclic immunofluorescence. Okay, so that really summarizes kind of the tools that are available, the assays that we run. But if you already have data that you've generated in other ways and you just want to see what they look like in GR space, there's an online tool. I think Peter also mentioned this morning at grcalculator.org, where you can import your data and it will output GR values for you and some plots. You can also browse existing data sets and play around with looking at different cell lines and cell treatments that we've looked at in the HMS Link Center. Okay, so that was quite a lot of information, but those are the methods that we use. Again, I can pause for a couple minutes if there are any questions about the experimental side of things before I wrap things up with just a couple slides on the takeaway messages. Okay, I'll keep going. All right, so again, looking at what we refer to as the MCF10A reproducibility project. So early on in links, we decided that if we're all going to be generating dose response data, we know that there are issues comparing these large dose response studies. We had better be sure that we can at least generate reproducible data across link centers. And so what we did is we took one cell line, shipped aliquots of it to all the different link centers, shipped aliquots of the same drugs to all the different link centers, as well as the protocol, and each center collected what was intended to be the same dose response data. And so that's essentially what's outlined here. We developed the protocol and collected several data sets. All the other link centers collected data sets as well. And then we looked at some of the factors that contribute to reproducibility or a lack thereof. And so within center accuracy was really good. This isn't surprising, I hope, but it was still, I think, an important sanity check that different scientists within one center can produce highly consistent data. But when we initially looked across centers, we were quite surprised by how poorly some of these dose responses agreed. Thankfully, by the time we finished, we were able to get all centers to agree very closely. I don't have a whole lot of time, so I won't get into it too much. Peter talked about it this morning, and you can reference the paper, of course. But one of, I think, the most important takeaways is that dose responses are not always interchangeable. And so as part of the exercise and part of the experimental design, one link center used the cell titer glow assay. And as you can see that in some cases, neratinib is a great example, the assays are totally interchangeable. So if you use a microscopy-based assay or you use an ATP-based assay like cell titer glow, you get virtually indistinguishable dose response curves. That's not always the case, and then the opposite extreme is palbociclib, where a cell titer glow assay would actually lead you to conclude that this particular cell line is not at all sensitive, whereas a microscopy-based assay would lead you to conclude that there's actually quite a dramatic effect. And so what's going on? Palbociclib induces cell cycle arrest, but the cells remain really highly metabolically active. They get really large in size, as shown in this micrograph. And so because cell titer glow is a surrogate measure and measures kind of metabolic activity, really, these cells are metabolically active. And so you don't get the cell count response that you expect, or that relationship, I suppose, is broken. And so these are the factors that were identified at the end of that paper as affecting reproducibility, and some kind of best practice recommendations for addressing them. They range from things like, randomizing your drug layouts, as I discussed, to making sure that your drug stocks are QC'd. STR profiling was mentioned. This didn't come into play for our study, but of course it's a really important thing to to be sure of, that you're treating the cells you want to be treating with the drugs that you want to be treating them with. And then some best practices for methods as well. So, running those pilot experiments to optimize your conditions, and using what we refer to as sterile data techniques as much as possible. And so that really just means using the automated tools that I presented, so that you can quite easily reproduce your analyzed data at the end of an experiment. And with that, I think I'm just under the wire. A lot of what I presented today was work that Mark Hafner did, and Kartik Subramanian as well. And then on the experimental side, the DyeDrop assays were first established in the lab by Mario Nepal, with a lot of expert technical help from Mira Chung, and then more recently from Kiara Victor and Ben Gaudio. And with that, I will stop and take any questions or open it up to discussion. Thanks for listening.